Hey, what is going on? This is February 23rd, 2022, and today is going to be a short video because I have a lot that I need to talk about with in regards to the cell processes and cell cycle regulation. I did mention yesterday that I would be talking about G proteins and G protein coupled receptors and things like that. And that's all important. But I wanted to step back for a second, and I will get into this a little later more in depth. But for now, let's just get kind of the ball rolling, and we can talk about the details in subsequent videos. But yeah, and by the way, I seem tired today because I had one tequila sunrise yesterday at a networking event, and that kind of completely uh, it's threw me off. Like I drank it, and as soon as I drank it, my muscles were like uh, they felt very like pained. I don't know. I have to look into why that is. But anyway, I will from now on avoid the tequila sunrises and get um, maybe a vodka sunrise. I don't know if that's such a thing, if that exists. But anyway, yeah. So I'm not gonna go into too much regarding my personal life now. So let's talk about um, the cell cycle and the cell checkpoints, right? So remember we talked about the growth phase of the cell cycle, that the synthesis phase and the growth 2 phase. They're called gap phases, but I call them growth phases because at one point people initially called them gap, and they called this because they thought that much wasn't happening during the gap 1 and gap 2 phase. Even when I was in school, I remember vaguely that people were saying that, and I mean like grammar school and whatever, that nothing really much happens during the gap phases. But that's known to be completely untrue, because these so-called gap phases are where the cell prepares to go into the S phase and create new genetic instructions, duplicate genetic instructions. It prepares for mitosis by dividing its organelles up and doubling them. It's so everything is in preparation for mitosis. And just to recall, like the, you know the transition boundaries between these phases, right? These growth slash gap phases and the synthesis phase are dominated by, or I guess they're directed by these control points on the cell. So what's important, and without going too deep into the details regarding the control points just yet, is that these control points have an fairly complicated processes happen here that are analyzed by cancer researchers. So it's important to know what's going on here. And I think a diagram like this probably, I guess, illustrates it better, because you have you basically have to see whether, depending on what checkpoint you're at, whether the DNA is damaged, right? And then if the DNA is damaged at a particular checkpoint, then the cell has to go repair it. Then you have to see if whether the replication, after it's done, after the synthesis occurs, whether the synthesis has faithfully replicated the DNA or not, which DNA polymerase, which I'll get to a little later, is pretty good at that. But over millions and billions and billions and trillions of DNA segments and several billions of nucleotide pairs, which the entire genome is copied every time, right? There's bound to be errors, or that even the DNA polymerase, millions of them, will run afoul of precision error, and eventually have to be corrected. And then finally, the replication checkpoint. There is another DNA damage checkpoint, right? So there's several checkpoints in the cell that are related to making sure the DNA was copied faithfully, because really if the DNA wasn't copied faithfully, then the risk of development of cancer is very high. And the reason why is because this cell cycle regulation occurs because there are proteins which are created, which are transcribed, which govern the regulation. And if there are errors in the proteins that govern the regulation, then the DNA, then the cell itself will not know or not respond properly to signaling events. Okay, so I'm primarily talking about cyclins and cyclin-dependent kinases, right? So there's like so many actors within this realm, and the way that I like to think about it when I'm looking or trying to understand what's happening in the in cell cycle regulation or in any cellular process, right, is the end products. Okay, so like for example, if you look at this and you're not familiar with any of these intermediate compounds or macromolecules, you will be very confused. And in this case, I think the best way to kind of like think about it is look at what's happening, right? Cyclin, CDK1, and you have protein kinases available. So let's just quickly go over cyclins before I go deep into cyclins during the week. And what I'm going to do is talk more about G proteins as well, right? So here is the cyclin expression cycle, right? There's a mouthful there. This is important because, again, the presence of cyclins controls the progression of a cell into another phase, right? So when cyclins are present to the right degree, right, and there is some thresholding that occurs here, but when they're present, because of their presence to a certain degree, the cell cycle can progress, right? And all the intermediate signals are given. For example, and you have these are the major cyclins. There's many different other kinds of cyclins here, but this is cyclin A, B, D, and E. I'm not sure what happened to C. Maybe it's around there. Whatever the case is, we'll go dissect an example. During the D phase, the cyclin D, the G1 phase, the cyclin D, right? So this is basically at the very start of a cell's life that hasn't undergone cellular arrest, right? Which is the G0 phase, which means it's not doing any of this stuff, right? It's not preparing at all for another mitotic event anytime soon. But in the G1 phase, preparation is still slow, and it can depend on the cells, whether they're labial cells or permanent cells or somewhere in between, the length of time that this is happening. But cyclin D production is going up, and it's kind of like circular, inverse parabolically going up, I guess, or no, maybe logarithmically is the best at this point, the best descriptor. And then what happens is, or square root of x, whatever, so we'll call it square root of x. So cyclin B goes up like the square root of x, or square root of time. And then what happens is that in the middle of the S phase is when cyclin D is at its maximum. Okay, and then cyclin D tapers off during the other phases until it drops pretty rapidly to 0 by the end of mitosis, right? And then again, one can imagine it will jump back up in the future again when G1 restarts for the new daughter cells. So cyclin E is produced between G1 and S, and ends somewhere. The production and appearance of cyclin ends at somewhere in the middle of S phase. And I want a cyclin E, and I want to like just be clear that the cyclins, right, you have now, we can't talk about cyclins without talking about cyclin dependent kinases, and this is a whole different thing that we can get into, but it's important because these cyclin dependent kinases, CDKs, right, what is their purpose? They are protein kinases. And if you remember what protein kinases generally do, they are responsible for phosphorylation, okay, or activation of proteins, right? So these are very core enzymatic elements in biology, right? Everything that leads to an action of something else is dependent on kinetic activity or kinase activity. And kinases activity, again, so you have what happens, you have a protein, just as a general kind of explanation of what that is, you have a protein, then you have a kinase which turns it into a phosphoprotein. What does that mean? this kinase complex comes by, which can be extraordinarily molecularly complicated. It comes by, and it's been in your blood for a while, right? These kinases are in your cytoplasm, cell cytoplasm for a while. They just kind of float around. The kinase will be, the kinase will take a phosphate group and attach it to a protein, turning into a phosphoprotein, right? And that's this phosphobinding protein is the kinase, comes by, takes a phosphate, turns it into a phosphoprotein, and then you have this example, like, will illustrate an example of that, but you'll have oncogenic pathway activation, which then allows like the production of proto-oncogenes and whatnot. So anyway, so these protein kinases are always available in your system, in your cytoplasm rather, but these cyclins come by and will then, depending on their presence, they will activate these kinases, and those activation of those kinases will then allow transcription factors to be released and created, and those transcription factors will lead to signals being created which allow the cell cycle to progress or to not progress, right? So it is in a way it's simple, right? Because one thing unlocks another thing, but in another way it's very complex. And when I go more into like the actual like pathways, because the chemical pathways in the cell, right, are the most important things when it comes to like determining what will end the chain of events that leads to a product that will progress the cell cycle. There's a lot of branching going on and whatnot. So we'll go into all of that. But for now, I thought I would just have this video illustrating or elucidating a little bit about the primary actors in the cellular growth processes, and we'll get into a lot more of that at another time. I'll see you around.